Hello, welcome to Converging Dialogues. This is Xavier Bonilla. On this episode, I'm very happy to bring the conversation I had with Alfonso Martinez Arias. Alfonso is a developmental biologist studying embryonic stem cells. He has his PhD from the University of Chicago. He also had a postdoc in Cambridge and was professor of developmental mechanics in the Department of Genetics at the University of Cambridge for many years. He's the recipient of numerous awards and is the author of the latest book, The Master Builder, How the New Science of the Cell is Rewriting the Story of Life. And that's what we talk about in this conversation. We start by talking about why many people today overemphasize genes and why there's less of an emphasis on cells. We give a kind of overview of the basic makeup of the cell. We talk about the role of gametes, talk about selfish genes and selfless cells. Talk about cells and embryos. We talk about embryonic stem cells, of course, and how, if they're necessary for human development. We spend a good amount of time in the conversation talking about the ethics in science and public communication, and specifically bioethics. And what are some ways of talking about science, obviously accurately, but also in a balanced way. We talk about polygenetic index scores, we talk about cancerous cells, and many more topics. I was very, very excited to talk to Alfonso. He's a great researcher and just a wonderful, wonderful man, and I really, really enjoyed our conversation together. As always, you can find this conversation and all other conversations at convergingdialogues.substack.com, also on YouTube, so get over there, follow, subscribe, and share widely. And now I bring you Alfonso Martinez Arias. I am here with Alfonso Martinez Arias. Alfonso, thanks so much for coming on the podcast. I'm greatly looking forward to talking with you.
Thank you, Xavier, for your interest in my writing and myself.
Of course. You've written a fantastic book, which is called The Master Builder, How the New Science of the Cell is Rewriting the Story of Life. It's got some wonderful insights here, so I'm excited to talk to you all about it. Before we get into the book and your work, just give me the kind of snapshot of who you are professionally and academically, and what you're currently doing.
I'm a developmental biologist. I was born in Madrid, and I studied in Madrid, my undergraduate. For my graduate career, I went to the University of Chicago in 1978. Where I studied, curiously, biophysics, but that turned. My great ambition was always to understand development, how one cell becomes many, and how many become an organism. So at the time, I thought that you could use principles of physics and mix them with biology to understand this. But sometimes one is too early, sometimes one is too late. But I think I was too early there. And that was the times at that time. And I think this is important to understand perhaps the trip that I've been lucky to go through over the last few years. We didn't know genes were involved in development. We didn't know the character of the molecules that underlie that marvelous transformation. And it was at that time when I was in Chicago that the revolution of molecular genetics started to happen. And I realized that was probably the way. So for my postdoc, I was lucky enough to be accepted in the world-famous Laboratory of Molecular Biology in Cambridge. And that was a wonderful time. I mean, when I arrived there, the genes that were associated with mutations in development, and I'm sure that we'll talk about this, were started to reveal some insights into the genes that were perhaps determining development. And for about 5-6 years, every day was a new discovery. It was absolutely exciting. Not that we understood what we were discovering, but it was really something new every day. I was very lucky to remain in Cambridge after the postdoc, first as a Wellcome Fellow for many years, and then as a lecturer, and then professor in the Department of Genetics in the university. And really the best time of my life. About 2 years ago, in the middle of the pandemic, I decided there was a very good opportunity in Barcelona, Spain, and I accepted the research position here. This is a hub of excellent biological research in Europe. Many people say that it looks like the Bay Area, but the Mediterranean is not the Pacific. So the thing it's good. But and here I am talking to you and looking forward to our discussion.
No, that's great. That's fabulous. You mentioned the kind of maybe too early. It reminds me of I talked with a biophysicist maybe a couple of years ago now. His name is Raghuveer Parthasarathi. He wrote a book called "So Simple a Beginning," and he kind of fuses this world of biology and physics and where that's at, and the whole biophysics. Wonderful person. He wrote a great book, and it's interesting seeing a lot of the intersection of all of these different types of disciplines coming together to, kind of help each other do life's homework of sorts instead of being
over the last 20 years, that time that I was looking for in 1978 has come true, and I've been lucky enough to collaborate and work with some of very good people in that field. And that's because biology has become quantitative. And as we shall discuss, because we need to understand the cell. And the cell is a physical object with very exciting properties. I've just realized that maybe, again, it's a bit too early for some of the ideas that I have, and I've had to go back and try to understand cells in their terms, not in our terms. But you're absolutely right. And right now, that intersection, physics, quantitative biology, cell biology is an amazing field.
Yeah, certainly. It certainly is. So let's talk about, I guess, the major thesis of your book. You make the case, and it kind of resonated with me because we've seen a lot of this, that there's an overemphasis on genes in many fields of study. Many people talking about genetics and overemphasizing how important genetics are, and genes are certainly important, but people do tend to overplay the hand a little bit. And I guess, how would you say that the cell is more of a central player, and how do you resist, I guess, maybe overemphasizing the cell, right? Some people may accuse you of saying, well, you're overemphasizing genes, but we don't want to overemphasize cells. So how do we see its importance and make sure that we have the right understanding of the marriage between the two?
I guess the quick answer to that question is, how much time do you have?
You tell me.
I think that you've posed a number of things, and I'll just go to the heart of the first one. I mean, why overemphasizing the cell? Well, when you want to be heard, you have to be loud. I think that this is a very, very clear case. I think I state many times in the book that it would be foolish to say that genes don't have anything to do with what we do. But I think there is right now a huge imbalance and a huge oversight even of some of the metaphors that are used about dealing with genes and us. When the Human Genome Project was finalizing the first draft, very big names, we have now the script of life. We have the recipe to make a human being. Well, there are two metaphors that are often used dealing with the genome and us or any organism, and I have questions for them. They say that the genome has a toolbox, is the toolbox to make an organism. My question is, who uses that toolbox? I mean, this is a very good thing. They say that there is a blueprint. Okay, who executes that blueprint? I have never seen the blueprint. I was lucky enough to spend a lot of time working in the prime organism to link genetics and development, which is the fruit fly Drosophila melanogaster. And it was in playing with this organism that slowly it became clear to me that the emphasis in genes, what it was doing is transforming what is a tool, genetics, into an explanation for a process, which is the development of an organism. And I think we have to be careful with that. I think I will overemphasize cells simply to call attention on questions that are there that need answering. And we should not get out of them by saying, oh, it's all in the genes. As I'm sure that will come out in our discussions, there are obvious issues trying to blame genes for things that they have nothing to do with. And as we shall also see, cells can do things that genes cannot dream of.
Yeah, tell us about that. Tell us about how are cells are essential for genes, and also what are some of the limits of genes, right? So to chat about that a bit.
Well, take something that has been very much in our minds over the last 4 or 5 years. Take a virus.
Mm-hmm.
A virus is nothing without a cell.
Mm.
It's just a piece of information. It needs a cell
Mm-hmm.
to hijack all its machinery to reproduce itself. That is a very good example. Many people will dispute whether a virus is a living organism or not, but what it is, it's a piece of DNA. And if you're going to tell me now that a piece of DNA is not life, then, I mean, the conversation doesn't go anywhere. But what is very, very clear, that even that very simple form of life, which is very mean, as we know, it needs a cell in order to execute all its processes. I think, of course, a cell comes alive during development because all of a sudden you have something that builds something. I think I like to see the genome as indeed a toolbox. It's a set of instructions, but instruction for tools and materials that now are going to be harnessed by cells. Now you can say, what is cells? A cell is a conglomerate, is an entity, which we are starting to understand right now, which results from emergent properties of its components. And I can give you several examples in which you can have cells living and working on 2 dimensions. And when you put them in 3 dimensions, they will do totally different things. It's the same cells and the same genes. However, there is information that they are able to process and to harness now in a very different way, to use the genome in a very different way. So, and I think the great challenge of the century we are in is to precisely understand what are those properties and to try to build a new biology. It is early days, but there is no question that this is what's happening. So genes on their own, they cannot do very much other than to, at best of times, produce information to create proteins.
Do you think that there was an explosion of understanding of the power of genes for many years, but people didn't kind of put together what you're saying, right? Like there's a bunch of tools, but someone's got to use them, right? And cells are kind of an active participant of genes. Why do you think people went so far with genes? Is it just the novelty of it that we were learning so many things we hadn't with technology? Or, why did cells not get as much of an emphasis for a while?
Of course, here we enter into a little bit of a historical analysis of a very important moment in history. We also have to remember that history is very long and we live in a very small period, and biology is a very new science. And genes have been, I mean, the discovery of the gene, the understanding between the relationship between genotype and phenotype, particularly the relationship between specific mutations and specific diseases has been very, very important to place genetics and genes at the center of our lives, because there have been some remarkable successes, particularly in that area of diseases and illness. However, when you go to the number of diseases that exist and the number of genes that exist, you realize that actually what we can explain, the number of diseases that we can explain with genes, is very small. In a manner that is related to genetics. But I would say that the emphasis of genetics is, on the one hand, as I have said, the relationship of mutations, which doesn't mean genes, with disease, and also, of course, the huge impact that the science of genetics has had on agriculture, for example. I mean, because it's really been very, very important. But I think we've got carried away with that and with the sequencing of genomes. And then, all of a sudden we start doing what scientists should never do, which is to explain everything in terms of this marvelous entity that we have defined, that is the gene. I think that is foolish in 2 ways. One, because you cannot explain everything in that manner, but also because by doing that, I think we might become blind to a totally new biology that might underlie there if we ask the right questions.
Yeah, so it's very, very important to kind of remember or recall when people are reading about genes, and how many people in certain fields will overplay this emphasis of genes and environment. And it's like, yeah, that's important in development, but there's a lot of other components there, namely cells. So tell us about development. You talk about it in the book. Tell us about the epithelium and how that's very essential for cells, and just kind of walk us through kind of the structure of a cell, maybe stuff that people will know in high school or undergrad, of the organelles and the mitochondria and ATP and Golgi apparatus and the nucleus, all that stuff.
Biology 101.
Biology 101. Right.
No, I'm not gonna expand a great deal. I mean, the eukaryotic cell, but the cell in general, it's a tremendous invention in evolution. I mean, as the late Louis Wolpert, a fantastic developmental biologist whom I was lucky to know, I remember once him saying, once evolution discovers the cell, it's downhill from there. I mean, and I do like that. Once you have a particularly eukaryotic cell, the possibilities are immense, because this is one of the things that we need to understand about cells, that they have a repertoire of behavior, of activities, of processing information that it doesn't exist in the DNA, and that we are just starting to understand. So what is a cell? A cell, as I said, when we talk cells, you and I here tonight, we're talking about eukaryotic cells. The eukaryotic cell, it's really remarkable. I like to think of it as a machine. Many of my colleagues don't like the idea of cells as machines, or that there is a machine theory of biology, but I do see them as machines of a certain kind. They have a nucleus. I mean, that's where the command center, if you wish, of the cell. That's where the DNA is. That's where and around it, encased in a bilayer lipid membrane, there is a universe. There is a world of activity. There is a lot going on, and we are discovering that in that going on, there is a structure. There are molecules moving at incredible speeds, but the thing is so condensed that there are cables that proteins walk through to move cargo from one place to another. The membrane contains receptors that listen to what the other cells or the environment say. There is machines that are dedicated to dividing the cell in two when it's grown enough to generate an offspring. I mean, there is toilets that the cell used to process the food that it has. It's a whole universe in there that we're discovering, and all, as we've seen at the end of the 19th century, when the first pictures of cells appear, some of this structure, many of them have been discovered, and they are represented as static entities. I think the vision that we have of the cell 120 years later is very different. It's of a very dynamic entity. Okay, one very important component in the recesses of the cell is this structure called mitochondria. Mitochondria are very important because they are the powerhouses of the cell. They are the ones that return. They look like bacteria. And actually, they were once bacteria. In fact, the secret, the origin of the eukaryotic cell is a big mystery. And famous Lynn Margulis made a proposal many years ago that the reason why mitochondria look like bacteria is because they were bacteria that were eaten by another type of cell. I mean, that was not accepted for many years, but now this is called the endosymbiotic theory of life, and really of the cell. And that is a marvelous story that before these very complex structures that we call eukaryotic cells arose, these bacteria existed, and there was probably a huge world of different bacteria, and another sort of kingdom, which is called the archaea, which are more, that we are starting to learn about, that are more interesting entities. And interactions, you can imagine a very exciting world in which everybody is interacting with each other, having pseudo-sex, if you wish, with each other, eating each other. And sometimes in these interactions, something happened that both entities understood that could be beneficial to each of them, and then a eukaryotic cell. One thing to understand about biology, because there is no, we can have sketched out a little bit the basic structure of a cell, but the cells are diverse, they are different. You know, a neuron is not like a red blood cell. An intestinal cell is not. During evolution, this is one of the big properties. And, you know, to contrast, DNA is always the same. is, and many of your listeners might forgive me, a boring double strand of information of ages, and this. That doesn't change from one cell to another, which is packaged with these wonderful numbers that we all like to quote. But cells have this ability to evolve, to generate different classes, to different. An epithelium that you make, I mean, you could say, in a broadly speaking, that there are two kinds of cells, some that are epithelial and some that are mesenchymal, as they say. I mean, this happens very late in evolution. We've made a big jump from this sort of entity that arises, something that we might turn to later again, is the origin of animals, another huge mystery, you know? I mean, how do you go from these single, sometimes colonial organisms that flood our rivers and our life to these things that we call animals, the most primitive ones probably being the sponges, of the familiar ones to our listeners. So that is another big jump. But once you have there, the cells start specializing, and epithelia are like, how should I describe it? It's like the bricks of the cell. They are flexible bricks. They are cells that have very well-defined organization. On one side, they have a set of proteins that allow it to interact with the environment. In the others that allow them to interact with other cells. For example, your skin is a very good example of epithelial cells. The lining of your intestine are very good examples of epithelial cells. And the construction of these epithelia are a very good device to keep different cell types separated, to create very flexible structures like it is your cells. And during development, during the building of an organism, there is a lot of transformations between cells that are epithelial to cells that are mesenchymal. And, you know, all this is using the genes all the time. Cells are going to the genes to draw the materials and the tools that they need to build this. But epithelia are probably a very, very good way of making very good use of space. I mean, that's what you can see, and to build the structures that we have that are very, very interesting.
It's interesting. It sounds like you were talking about the double helix it feels a little bit more static, whereas cells can have so much dynamic qualities where they're moving and they're changing and they have specialties and they're doing different things all the time. And I was curious when I was reading the book, when you were explaining all these things, and I was like, okay, a lot of this tracks, but I guess one question I have is about meiosis and gametes. Now, you know, I won't get you in hot water. So, but gametes have become more talked about for biology, about what it means to be male and female and things like that. I guess, how do we understand this process, at least for humans, and maybe just for all multicellular organisms, of gametes and being essential for understanding what we have commonly known as biological sex, and meiosis, and how is there a lot of dynamic qualities to that, where there's a kind of changing and moving and evolving, or do these stay relatively static, and the role that the cell plays with these parts?
Well, I think there is no reason to call hot water. We are talking about biology. You are asking me a question, which, if I understood correctly, is where do gametes come from? What are gametes? Yeah. I mean, that's very, very simple. I mean, you know, in that dynamics that cells have, there is reproduction. I mean, this is a very important element of life on Earth. Yeah. And a lot of the prokaryotic cells, which we can put there, the bacteria, they and a few other related organisms, they divide by scission. There is not a lot of sex. Now, what happens with this kind of reproduction is that there is not a lot of variability generated. I mean, you're going to become different because at the bottom, you have this repertoire of tools and materials that is in the genome. The only way that you can generate novelty is by mutation that will happen and that will be selected for. At some point in evolution, sex is discovered or invented, as you wish. And that is clearly one of the big problems that evolutionary biologists have. I am not an evolutionary biologist, and that would be getting into hot water if I was to speculate on the origin of sex and what that happens, largely because of my ignorance of the subject. But every time I try to penetrate, basically nobody agrees, and there is lots of theories. I think this is one of the problems of biology, that very, very often during evolution, the path that leads to us is a sequence of unique events, as somebody called recently a series of black swan events that we cannot. It's very difficult to reproduce. It happened, and from there on, one of the examples, and don't forget, I'm not forgetting about the gametes, I'm not going off tangents, is the origin of life. Personally, I don't find that a very interesting question, because I think there is so many ways in which it can start it, and we could spend half an hour in which I could prove you that well, I'm very glad that people have views, but again, this is probably a black swan event that happened and things. So the same with the origin of sex, but it is clear that led to the formation of gametes, which are the sexual cells, and there is people that can explain why to have two is better. But the fact is that when we have that, there are many mechanisms all of a sudden of generating variability. Because one of the things is that most of the cells on the face of the earth, they are diploid. That is to say, that they have two genomes that come from each gamete. And in fact, the way to make the gametes is that you make sure that these cells now have only one genome. And the genomes are different. And they've jumbled up in the process of making the gametes, such that when you get to individuals coming together, you're now getting something new that didn't exist in any of the parents. So I think that that's the evolution of those cells, which are very sophisticated, are very complicated and interesting. The sperm, that's why when Leeuwenhoek, one of the person that discovers that uses the lens that he built to start looking at nature, when he puts his own semen under the lens and find these things, he thinks they're animals. He cannot relate them that they are cells, and then it took them years to find particularly the mammalian egg, as I also discuss in the book. But these are very, very special cells, each with their own characteristics. And when they combine, when they come together, they generate an entity that is totally different from any of the parents. Otherwise, you have clones. So I think gametes were clearly a very important evolutionary moment, because although nobody can explain why it happened, and you'll see many discussions about their advantages, the fact is that they are the basis of what life is.
You have a nice phrase in the book. I don't remember where it is, which chapter or whatever, but I believe you say somewhere in there that we have selfish genes and selfless cells. Right. It's a very nice phrase. So just tell us about what you meant by that.
So that is embedded. We're jumping a bit about, but I'm following your lead. I think that's very important, because and you're going to forgive me and allow me to expand here a little bit, because this is important.
Yeah, please.
I think that the culture of the gene, let's call it like that, which is very, very important, and it's played a very important role. As I keep on saying, I'm not saying that neither the genes are not important or the genes, it's clearly, but there is another entity that plays a role here as we are developing in this conversation. One of the very important and iconic moments in the history of the gene in the 20th century is the publication by Richard Dawkins of The Selfish Gene. That is a very important book that has an idea which is like a climax of this gene-centric view of the world. One thing that is very important to understand, Richard Dawkins is an evolutionary geneticist, is an evolutionary biologist with a tendency for behavioral ecology. That's probably that is his specialty. And this book puts in the minds of people the centrality of the gene in our lives and in evolution. Basically, his point is that the only thing that matters is the genes. In fact, he puts a metaphor which struck me as a graduate student, and it's a very striking metaphor, that we are machines built by genes in a sort of eternal fight that they are between themselves. I mean, this is the metaphor. I'm probably embellishing it a bit, but that's really the premise of the book. Genes are selfish, they're fighting with each other, and we are their product in order to, and as a behavioral ecologist, I mean, when you start reading his books, not the Selfish Gene, which is a rather readable book, but he has another which he wrote to explain to the scientists the selfish gene, which is called the extended phenotype. I mean, I hope you like aphids and little insects, because it goes forever about the multiple strategies of these animals. And he always blames genes. This is all the working of genes. And there is a problem there, because we are not a direct consequence of genes, whether we like it or not. I mean, I think also something that is very important to understand is that throughout the second half of the 20th century, there is a little bit of a dissonance between the evolutionary biologists, or most evolutionary biologists, for whom a gene is an allelic frequency. It's a set of allelic frequencies, and alleles are the variations of the genes, and that's what they want. And with the discovery of the material nature of the gene, there is the people that study cell biology, developmental biology, for which increasingly a gene is a real entity, which is transcribed into RNA and translated into proteins. And these are two very different views of the world. So the relationship between the genotype, which is the genetic content of a cell, and the phenotype, which is what we see, the function, the structure and function that results from the combination of genes, in the case of the molecular and cellular biology, requires a set of steps of increasing complexity, and we can use that word emergence. Whereas for the evolutionary biologists, there is an arrow, which is filled in no different ways. So this creates an interesting problem. A good friend of mine, Denis Duboul, wrote some time ago a very one-page article in a journal in which he poses what I think is a very profound and interesting question about how to bring together development and evolution. As I said, I'm interested in the development of an organism. And he puts it that it's difficult because development deals with a recurrent theme. That is to say, the development of an organism will recur itself over and over and over again. And evolution is really an arrow that moves through time forward without stopping. And there is a challenge there to bring together these two very different entities. Discussing with him, I realized that this is a problem of time scales, and that it is true that the evolution is really this arrow that is moving in time. But development is not totally inert. I mean, over time, it also changes. This is how we get evolution. And then you get this very interesting thing, and thinking about this in the book, I think we need to resolve this paradox that my friend Denis Duboul poses, but also try to fill in the gap that Dawkins puts in there, because Dawkins doesn't know any cell biology and doesn't know any developmental biology, and I don't blame him for that. I know very little evolutionary biology, but sometimes, and this is true in the extended phenotype, when he tries to fill this gap, it becomes obvious that he doesn't understand these things, because during development, the cell is ruling the genes. Cells are delving into the genome, picking up the genes that they want, using them to build new cells, different cells, to organize the cells in space, and they are collecting these sets of genes. However, he's right that in terms of evolutionary time scale, what matters to the genome is to be passed to the next generation. And this is why I suggest in the book that it's a metaphor, of course, but I think one is allowed to do that the cell and the gene reach a Faustian pact, in which, if you wish, the gene is playing Mephistopheles, if you wish, and the cell is playing Faust. And the cell sort of says, all right, if I can use you during my lifetime to build these wonderful things and to use my imagination, I will package you into the gametes and send you to the next generation. And the genome says, that's fine, Then I'm going to remain quiet. As long as I can get to the next generation, I'm going to remain quiet. And now you can do with me whatever you want. And I think this all of a sudden is a way of sort of trying to bring together the sort of, and the cells, when they start doing that, they are not selfish. They are helping each other. All of a sudden, they are controlling completely the genome. I would say that situations like tumors, like cancer, that are examples in which now the genome rebels. and tries to take over the cells. And now what you get is chaos, because it's trying to do what it does in the context of order and under the control of the cells. So this is what I mean. The cells are selfless because now in this entente that they have got with the genome, they are trying to build something over a defined period of time. This is also why I call it Faust, because if you think this is the myth of Faust reenacted in a way. And I think there is no escape. That toolbox that people talk about, I don't think there is a blueprint. The blueprint, the notion of blueprint is a very interesting and fascinating one. I think biologists, unlike physicists, and I've stand around many physicists, I think we are very loose with our language, maybe because the entities that we try to capture are very difficult to define. But I do think that this is one of the big problems of biology, that we use language with too much freedom and sometimes get into trouble, because as I haven't seen a blueprint in a genome, and I look for it, or a toolbox. And so there is all this information that is being used by cells together with emergent information, which is a very elusive phenomenon. But this is what I mean by the cells being selfless, but also of how this notion brings together development and evolution in a manner which, for the moment, I find comforting.
That's a nice way. The Faustian pact is a good one. It's a nice way of showing not genes or cells as in necessarily conflict with each other, but how are they working and coordinating together? When you look at an organism, and then when you look at humans, that reads differently, because I find that when people do emphasize one or the other, it does tend to be pretty myopic in how people are looking at things, and people will do this many times, maybe unintentionally. But you displaying kind of this interplay between cells and genes, I think is particularly instructive, because it helps us to kind of contextualize a little bit better what's going on within.
But you have to look at it in the context of these time scales, that they are looking at very different time scales. I mean, in a way, in the evolutionary time scale, the gene probably is the master, if you wish.
Right. Yeah.
But in the short time space of development, it's none of their business. I mean, they've been packaged into these
right.
units, the gametes, sent to the next generation. Now, for the rest, just play with me. And that's very important, because, of course, that also creates the opportunity to explore with new mutations, with new possibilities, with new activities.
Yeah, certainly. So let's talk about embryo cells and embryos. This is a cool part of the book, middle part of the book. I want to hear your explanation of how do we get from a single cell to a full embryo, and then obviously a person eventually, if successful. You must know the work by she wrote The Dance of Life. Drawing a blank. Zarnica.
Zarnica.
Yeah, Zarnica. Yeah, that's who it was. She wrote a great book. She was recently in the news. I think she had some kind of paper with a group of people or something like that. I didn't follow it too closely. A little bit. It was apparently somewhat groundbreaking of sorts. But she writes a lot about it. For listeners, she writes a lot about embryos and the single cell becoming a human being and things like that. So you can talk about her work or agreements, disagreements if you want. But how do you usually understand a single cell getting to a full embryo and that process? You can keep with humans, or you can use anything else. But how do we get to that point, from one point to another point? It's such a huge transformation, and I'm curious for your thoughts.
I think this is where cells come into the limelight, because this is the work of cells. It's not the work of genes. I think that sometimes science works in mysterious ways. I mean, when I told you that I went to Chicago with the idea of understanding this process, because at that time we didn't know very much about the genetic and molecular underpinning of biological systems. I thought maybe you could do it by thought. I mean, by the same way you build physics. But I think my eyes were open, and slowly we build this picture. I think very important there was a lot of what it's called experimental embryology before the 1950s. I think that was not surprisingly, that did not have a cellular base. I think people were looking at tissues. They were moving tissues around. They were playing with groups of cells, but the cell view of the world was not there. That sort of falls into a hole for about 20 or 30 years because people get tired of, as Spemann said, and Spemann, the very famous German embryologist, I've spent my life putting an embryo into ever more embarrassing positions and asking it to get out of them and seeing how they do it. And I think people got tired of cutting and pasting pieces of embryos, and because they needed to understand something. I think the thing that develops in the background is mutations that affect the development of an organism. These were collected as curiosities by people that work with flies. People, of course, that work with cattle, they knew that when they fed in certain things, they got cyclopic or mermaid-like observations. So clearly there was something going on underneath. It is in the 1970s and the 1980s that systematic screens for mutants that affect the patterning, particularly of the fruit fly, that reveal that all of a sudden you can, I think this is very important, that you can mutate a gene, you can mess up a gene and you will develop a certain phenotype, a certain abnormality in a very reproducible manner. Somehow there is a link. Now, what is very important to understand is that what we know often is what happens when you mess up the gene. It is much more difficult to go forward and figuring out what the gene does. This is where I think all of a sudden we have fallen into turning what is a tool into an explanation. But we can come back to that later. Now, it is at this moment that all of a sudden this mysterious process that happens in which the first cell becomes an organism, we start attaching genes through mutations to all these particular processes, to all these events. And this is really where we have been for the last 30, 40 years. I mean, this has been, I've witnessed this. And so the way people tend to think that process happens is because you switch on genes on and off. You switch cohorts of genes, and genes will turn on genes. And if different cells have different genes, they will turn on different genes. And in this sort of progressive manner, you get every cell to express different genes. The problem is that, as I've been saying, genes turn on genes that turn on genes, and the genes are different in different cells because they express different genes. But how do you get shape? How do you get form? How do you get time? How do you get all this out of things? In fact, if you think about it, back to the idea of DNA, RNA, these nucleic acids, I think that you find that the DNA is very samey. You can get these gene regulatory networks, and they're all going to be one like the other. But again, genes code for RNA, RNA will code for proteins, and all of a sudden these proteins, in the context of a cell, and I will come back to why I'm making a pause here, these proteins acquire properties, they're ensembles that they don't have as individuals. This is where the notion of emergence starts happening. Also, one very important thing, the cell, this eukaryotic cell that we referred to before, one of the things that is very important for Dawkins is the notion that the DNA is a replicator, that it has the ability to replicate itself. In fact, I will not argue with anybody if somebody tells me that the DNA, if the DNA is a blueprint for anything, it's for another molecule of DNA. There is no more than that. Now, do you think that the cell will appear out of these processes of mRNA, of DNA, of proteins? The cell is also a replicator. If you remove the membrane of a cell, the cell cannot make that membrane again. That membrane, wherever it's involved, is a total entity of the cell that it has to replicate together with the DNA. So there are other replicators in the cell that cannot be built de novo, that the DNA cannot build by itself. Okay, this is quite important. And during development, this happens. all the time, very, 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 very orderly. So I think that the way we understand now this process is in terms of genes, turning on other genes. Now, that has to change, because what we are seeing is that cells can sense a space, can sense their position, and can turn on genes, not depending on the genes that they express, but on the position or the physical environment in which they are, okay? And this is something that now it's the subject of very important investigation. And this is going to be they use sometimes proteins to detect that. But remember, this is not anymore simply a chemical process. There are physical inputs into the situation that we have. So how does a cell become an organism? It's a progressive sequence of events that we are starting to have the tools to ask that question. We know the components that the cell used to build two cells. When they build two cells, those cells now start interacting with each other and talking to each other with languages that we're just beginning to understand to build four cells of the right organization, and so on and so forth. We still don't know what that language is. I like to say sometimes recently to the students that, at the beginning of the 20th century, we didn't know anything about genes. Mendel is about to be discovered. We had all sorts of wacky explanations for this problem of development, okay? And I like to read about some of those wacky explanations because some of them are very, very insightful. Now we discovered genes and we start explaining. At the end of the 20th century, we can explain everything with genes. Now, when you do experiments, you realize that there is another dimension there. And I think it is our duty in the 21st century to find that entity. I think that human development has been a holy grail of developmental biology, but the foundations have been built on other organisms, on frogs, on flies, on fish, on. I think over the last 5-10 years, the attention has started to be on human beings. I mean, this is very important. The reason for this has been the embryonic stem cells that you have that are very important. And I don't want to jump, but I think this has become a tool in our arsenal of experimental ways of addressing questions, because these cells have abilities to organize themselves into structures that we call embryos that we had not thought before. It's very, very surprising, the ability that these cells have to, without touching anything of their genomes. We don't deal with their genomes. We don't deal with their genes. In fact, what we do, which is very surprising, is we use signals that the cells use to talk to each other, but we talk to them through those signals. What is very clear is that we don't speak that language very well at the moment, because what we are doing is not perfect. But
is there something special? I don't say special, but is there something unique, I guess, about embryonic stem cells as opposed to maybe adult stem cells or other types of stem cells? Like, what is it about? Is it so dynamic in early formation that makes it more malleable, or is it something else that makes it very powerful for other uses, embryonic stem cells?
Embryonic stem cells have the potential to generate the whole organism. Okay, this is what it is. In our body, there are stem cells. For example, the intestine, basically every 2 or 3 days, you get a totally new intestine because of all the wear and tear that is going on there. Every 2 or 3 days, you get. Why? Because there is some stem cells at the bottom who keep the potential to regenerate that intestine, and they do it all the time. I mean, the skin is every couple of weeks. I mean, this is but those cells are what they call, the potency is restricted to a particular organ. They self-renew themselves. There is a magic there that they can reproduce themselves, and upon need, they will generate the differentiated cell type so that we can continue to digest food or to do the thing of the. Think about it. Every day, your body produces 200 million red blood cells. In fact, I mean, this is very surprising. I mean, and that's because there are stem cells churning numbers there all the time, but they are very specialized. Embryonic stem cells don't exist really in the organism. They are a creation of the experimentalist. There is a stage in the development of an organism in which that potency can be captured ex vivo. Okay, this is very important. We have found ways. I mean, those cells do not exist there, but somehow there is something very special about them that a group of people it's a wonderful story. I tell about this story in the book. Just through a series of interesting accidents, they find these cells, which on the dish, they can sort of renew themselves forever without losing the ability to generate an organism. Okay, and this is what is special of these cells. Now, some of us realized a few years ago. I mean, the question that interested me and made me switch from the fruit fly that I was working and learning about genes and getting uncomfortable with genes to working with embryonic stem cells is the fact that I never understood why these cells, these embryonic stem cells that you could have in a dish, and in a dish, playing with chemicals, you could get them to differentiate into any cell type of an organism, but they would not make an organism. Now, if you put them into a very early embryo, they would contribute to the organism. So there was some magic here. I mean, either we were missing something, or there is something magic about the organism that they need, or we were missing something that we better tried. And it is then that we started together with a small group of people to try to understand if we could sort of coax these cells into doing in vitro what they did in vivo. But these cells do not exist as stem cells in the organism. Unlike the intestinal stem cells or the skin stem cells or the blood stem cells, there is something very special about these cells, which also they have to do with mammalian development, and it's quite interesting. But I hope that these are very, very special cells. I should say that these cells also age. So after a while, they stop really doing very well what they have to do. Not surprisingly, but nothing lasts forever.
Maybe we don't understand all of the ways or reasons, but it is essential for human development, embryonic stem cells. You can't develop as a human without that, or no?
That's an interesting question. Let me phrase exactly what you have asked me, because it's a question that I had never heard before. Are the embryonic stem cells essential for human development? In the embryo, these cells exist only for a very short period of time in the early embryo. And indeed, without those cells, you will not develop. That's because they are the progenitors of all the different cell types of the organism. What they don't do in the organism, you don't keep a reservoir of these cells forever. Okay, these cells are there for a very short period of time. What we have done, what scientists have done, and some of us have learned from them, is to culture these cells in such a manner that they don't lose that potential, which is remarkable if you think about it. Absolutely. It's remarkable.
Well, I mean, this is where people, when this came out, I don't know, 20 years ago, and we started seeing this and said, well, maybe we could use this to help maybe cure or fix or alleviate certain diseases, right? That's the whole thing of it, right? Is potentially the power to help with things like neurodegenerative diseases, right? Alzheimer's and Huntington's, right? Isn't that the whole theory, right?
I'll be very careful in what I say. I have a tendency to get into trouble, and I'm doing very well today. You haven't realized, but you've provoked me. But I've done very well so far. I don't know how long will this work, but I think that the reason some of us get into this or are interested in these questions, particularly with me, is because I want to understand how cells build organisms.
Right.
There is no doubt that there are health and social benefits behind that. But by making that the prime target of what we do, we're not gonna get there earlier. I think this is a problem that the industry of stem cells, whether it's embryonic or adult, has at the moment. I think that there are things that I openly despise about this business, and one of them is precisely the abuse of the sensitivities of the people to further one's agendas and one's careers. Because sometimes when I hear some of my colleagues say that they're going to achieve all these things, either they are more stupid than I think they are, or they are lying to the people. I mean, that's the only two possibilities, because this is at the very early stages of this work. There is no question that there is, over the last few years, a number of very important investments have been made on the idea of using stem cells to cure diseases. Doug Melton, a developmental biology at Harvard University, with a company, has spent the last 20 years to try to get insulin-producing cells from embryonic stem cells. It looks as if he has achieved that. Now, this is not a simple matter of making these cells that will do what you want. You want them to do that in the context of an organism. He has to immunosuppress the patients. It's taken 20 years. When you think about the amount of money, probably trillions of dollars that have been invested in this project by different people, and that only now we have what is a promising but tenuous sort of result, you can see that anybody that says that they're going to start a company to cure neurodegenerative diseases from embryonic stem cells, it's really, you have to look at them. I understand how companies work, I understand how Silicon Valley works, I understand how business works. Sometimes it's crossed my mind to with our work to do those things. But frankly, I'm not in the business of BS. I'm in the business of understanding how cells build organisms and build. So I think that this is something that sometimes creates a cacophony in the field that is not healthy. And in fact, you did mention Magdalena Zernica-Goetz, and this is one of the things that gets me, that I think is problematic. She uses the media a great deal in order to promote things that we as scientists know are not what she said. And these headlines to which you refer to, as I said, I will not dwell on it unless you want me to, but I think they were very dangerous and have created, because at first, when we came to see the evidence for what she had presented to the press without any evidence, turns out that it wasn't there. But this is something that she does over and over again. This is common in the field. I don't know why the stem cell field tends to attract this kind of activity, but I don't think it is good. Why? Because they are very sensitive issues, and because the public then gets afraid or get excited. The public is the one that pays our research, so then we get backlash. And a mistake or a bad statement can cost us a great deal in this field.
Listen. I agree with you. I think it's very similar to what happens in the social sciences, but with the physical sciences, the stakes are higher in some ways, because in many ways, I fully agree with you. One of the things I get very nervous about, and sometimes people can do it unintentionally or by accident, and some people have more pernicious intentions. Hard to know, who is who there, but is they have a finding. It's not replicated enough. We don't know the efficacy or effectiveness of it. And then we start talking about it very confidently, publicly. And then if it's wrong or it changes or it needs to be corrected, people don't like admitting publicly they were wrong about something. So they double down. And then they say, well, this is, of course it has to be this. And let me fix the data a little bit so that way, continues. And this has happened in my field in psychology. It's happened in sociology. It happens in economics, I guess. But in the other fields, it happens in people's, in my world, I spend half my time correcting people for things that are, bullshit. People talking about all of these things that we know that doesn't exist, and then you have to go back and then correct for it. But in genetics and biology and all these things, it feels the stakes are higher because people are always complaining that, this is playing God, or you're pulling this or that.
Exactly.
It's all of those things.
And I think also by the field that we are talking about is a very, it's a small field. The experiments are difficult. Not many people can work, but there is a group of people that are working with that. But only one person speaks to the press all the time and talks about her results. So what happens is that she monopolizes the impression. And when we have to explain what works and what doesn't work, and why the hopes that have been raised are not there, this creates a problem to the field, because the newspapers, it's happened to me with journalists. When you explain to them what the reality is, they get disappointed. That disappointment results in that they don't want to explain to the people what's happened, but that has raised the expectations of the people. And in this field, we are playing not only with disease, but also this person likes to play with they're going to solve the fertility of women, for example. Now you're playing with very sensitive issues here, where there is a lot of yearning. For you are talking there with huge feelings and sensitivities. Now, as I said, this is not new in the field of stem cells. The idea of making a human being is sort of the ultimate, but there is an interesting stream of characters over the years that have played on the themes and fantasies that can be generated from stem cells to make claims. And there is no doubt. This is the other thing of this interesting phenomenon, that what many people want is to be the first.
Yeah.
So they know that it can happen. If it's happened in your dish, in a corner of your dish, somebody else will do it. So why not reporting it? And you've put, it's like the Spaniards when they arrived to America and put a flag there. Didn't matter who was there. We'll deal with that 300 years later. But this is something that I feel very strongly about. I feel maybe some people might accuse me of maybe having a very strong ethics, if you wish. But I think in these times in which we live, where people talk about post-truth world, where people like Trump or Boris Johnson have shown us how far you can get with lies or misrepresentations, or call them whatever. I wouldn't like this to come to a science. I think that this is, I accept that we cannot get rid of people that will play the game like that, but I think we cannot fall into the trap of doing that. And I'm afraid that in this field of the stem cells, whether it's for cures or for reconstructing elements of human development. But you see, you mentioned some of the things before. The surprising thing of this event that you mentioned is that the journalists never check with other scientists whether this has happened or not. But now it's established. You see, it doesn't matter. It's a bit overdue. And I think one of the things, all this conversation, this fragment started with you asking me, why do people do this? There is no question, Xavier, there is no question that in the future, what we are developing now will be used for improving women's health, for improving fertility, and importantly, for regenerative medicine. But we have to create a very good foundation. And this is what a lot of us are doing. It's a very important field. It's interesting these days, people are talking about the Oppenheimer movie that is coming. And they're talking about, I am the destroyer of worlds. Well, physicists have been the destroyers of worlds. We can be the creators of a new world, but we need time. In fact, I always think that I like to say that we've been doing better than the physicists, because the physicists created all the rules after the event, after they saw the destruction. But for example, all the regulations of embryo research come before embryo research was possible, because in the discovery of in vitro fertilization, they realized that the world laid ahead, that perhaps they should put rules to. This is the 14-day rule, for example, that you might have heard of, the limits of culturing embryos in vitro. So I think there is an interesting parallel there, that the biologists have been doing much better than the physicists, and I don't want that to be broken. This trend that we have developed.
There was a great book that talks sort of about the history of this. I had a conversation with him with Matt Cobb. He wrote this book called As Gods,
Mhm.
and he wrote this history of biology and genetics and all of the things where people were saying in the 50s and 60s and 70s and into kind of modern day. Wait a minute. Wait a minute. We need to put some pull the brakes here on some of this stuff. We're getting way too ahead. And he talks about many of the experiments that have happened in Asia where they tried to clone animals and all of these other things and trying to do a full embryo and what are the when there's no rules or there's not an emergency brake, and people just go with it.
Well,
Sometimes you let things out of the bag, you can't put it back in the bag. I
well,
mean that
no.
that happens.
What we have learned, we cannot unlearn. This is a fact.
Right, that's right. Yeah.
And I think the issue, for example, you probably know the case of He Jiankui a few years ago, who all of a sudden claimed to have done this marvelous experiment. I think these people that make announcements without evidence, and when we see the evidence, is not what they propose. I think the sad thing is they're all gonna be there all the time. We cannot, because that's human nature. But I think that they don't help. I think they don't help the work of others. That also they, of course, affect in a strange kind of way the livelihood. Because how are you going to say, well, if that person has done that, how come you haven't? So why should we fund you to do something that somebody else has done, even if that other person hasn't done it? You see the problem with that. But I want to stress that this is a very exciting moment in biology.
Sure.
And I think that we understand a lot of how genes work, and CRISPR is a fantastic tool that allows us. But there is one clear thing, that the amount of disease that can be treated with CRISPR is very, very small, because it affects sort of the screws. However, let's not minimize or underplay the importance and the value of that. I mean, that's very important, because the life of any single human being is worth all the effort in the world. The numbers, it doesn't matter. However, we have to face the reality that a lot of the things that we might be interested in are not the result of one gene, they're the result of the activities of cells. And for example, one of the most interesting developments in biomedical research at the moment is CAR T cells that you might have heard about, in which you reprogram a cell in order to now sniff a tumor and kill it. Now, what you're doing is reprogramming a cell. You're not changing anything. You're letting the cell do what it does. You're adding more tools to its kit. So imagine the day that we'll understand. I mean, one of the problems with this regenerative medicine that we were talking around the edges before is that we still don't understand. We cannot control what these cells do, and we cannot get them to do what they do normally. This is what I meant by we don't speak their language normally. When we do these experiments, we use cocktails. I like to call it 20th century alchemy, 21st century. I forget that we are. Although as a very good friend of mine always tells me, Alfonso, don't worry, remember, chemistry is alchemy plus numbers. So I'm always hoping that if we put the numbers into this alchemy, we might get to speak the language. But it's very clear that the signals, what we put in the test tube, is exactly the same signals that the cells are using to talk to each other. So I think this to me it's where a lot of the excitement comes at the moment, in that we have to learn this language that is about genes.
Yeah, it's like what many people have a lot of the concerns, and I can see it. Some people have different opinions on this, obviously, but this is a polygenetic index screenings. Where you have a polygenetic index, you're doing various GWAS studies, and you're saying here's your polygenetic score. Obviously, with IVF stuff, people do the pre-implantation genetics, right? The PGTP, and people, it's consumer eugenics, right? It's like, how do you want to build these things or whatever. And I think in a lot of ways, I find history really important here, because I'm with you. I'm very, very much a cautious person, because of history of there's a lot of potential and power, and we should absolutely study this stuff for sure. But when we export it too quickly without knowing all of the things, we just repeat the mistakes that we did in times past in history, whether it's with engineering or technology or physics or biology, we have to be able to say, wait a minute, how do we build parameters or contours around this, which might be there's a push-pull there, right? Because you don't want to be too stifling, but you also don't want it to be the Wild West either, where things have no limits. And I think we're going to keep figuring that out.
You've touched upon a very interesting situation that is developing, which is this polygenic risk-course.
Yes, yeah.
Which is really, I'm actually quite concerned about that because, and I'll give you some examples, but the problem with the polygenic risk-course is that there is a lot of money behind that.
Yeah, oh yeah.
This is where the problem lies, and really, what the polygenic risk-course, the geneticists that perform those studies, I think sometimes it's as if all of a sudden I were to start talking Latin to you, you would be very impressed, even though you wouldn't understand anything or some deep European language. You would be very, and you would say that I'm thinking something magic. This is the problem with these algorithms that try to calculate the risk that you might have or the heritability of a particular trait. When actually the numbers are very, very small. So what they do, if 3 genes do not give them enough numbers, enough statistical power, they will make it 5 or 1,000. And now there is people talking about not the polygenic risk-course, but the omnigenic risk-course, which is to say, if you have a problem in your heart, your whole genome has to do with that problem. Thank you very much. I think this is actually a very good point to highlight how we need to represent the cell in a different way and understand because what we are looking is at cellular diseases. We could represent the computer upon which each of us is working on in terms of Schrödinger equations, but that would be useless. What we are talking is about higher level sort of understanding, and this is what I think this polygenic risk-course is doing. The whole thing of the baby selection from polygenic risk-course is absurd because there is not enough variability there to them. But what there is in a lot of these situations is money, and this is what is the big problem here, and the ignorance of the people. I think this is also where we need to, as a scientist, I feel that we have a responsibility to explain what we do in no uncertain terms, and the limitations. And this is something that people are often not prepared to accept.
I agree.
That maybe there is something that we don't know, but we forget to add yet. Yes. Something that we don't know yet. Yes. And history has shown us that yet is very important because it will be covered.
I fully agree with you, because we had this conversation and we still have this conversation around COVID and the vaccines. And again, this is slightly tangent, but sort of related in this idea of when people are trying to talk about a novel virus and trying to find in real time with a moving target solutions for that for billions of people on the planet, you're going to get things wrong. And that's what I wish that people that were leading the charge in that is that they would have said that a little bit more clearly. But that doesn't invalidate the things that they had said, or that doesn't invalidate the efficacy or the effectiveness of vaccines, or it doesn't invalidate those things because somebody got sick. I think there could have been a little bit more of saying, like, we got this wrong. We thought this first. Here's how we've changed it. But, and you see that with other things too. Everyone talks about AI and all that. We have to be able to say, like, we don't understand all of this. We got some of this right. Some of this we still need to work out. And that's why you do good science. I think sometimes our arrogance and pieces of our personality get too in the way of that. Sure. And then it becomes a problem. It becomes a detrimental problem, because now you have people doing their own research on, they read an article, they saw a YouTube video, and everyone's a virologist, everyone's an immunologist, and it's it's quite bonkers to me. And these people get incentivized and platformed and all these things, and that's dangerous to me. And I don't think we should censor
Well,
people, but we
I
need to
think
combat
I
it
think
with
we
better
live.
ideas.
I think exactly. I think we live an honesty. I think
Yeah.
we live in a world. Many people see the devil in social media. Many people see the devil, but I'm afraid that this is the reality in which we live.
Yeah.
It has its good things and its bad things. It depends on how you use it.
Agree.
And I think as a scientist, I think our role and in the areas that pertain to me, I think we need to help people understand what is that we do, why we do it, and how we go about it. And I think the issue of the pandemic, I lived through it in the UK, where, as you know, there was a lot of doing and throwing in a very different way from the States between the scientists and the government. I think, the scientists should have been heard more in many different ways. But I think we live. This is what we were talking before, in a society that prefers headlines to detail. I think this is where we need to find a way to reach the people without, and in a way, if you wish, this is what I've tried. As I say at the end, this book that I've written, it's a way to try to put on the table a reality, which, if you look around, you will not find many books about cells in your local bookstore. I mean, if you go, you will find lots of books about genes. Everybody has something to say about genes. The sociology of genes. In fact, the book of Siddhartha Mukherjee, The Song of the Cell, is pretty much the only other one. And the book, which is very, very nice to read, like everything he writes, it sort of tends to look at the cell more in terms of its internal machinery, and it can go wrong, how it can go wrong. I like to highlight this more, if you wish, from the outside mystical properties of cells, but science is about debunking mysticism. It's about putting the gears. And if you think about the history of astronomy, if you think about the history of anything, really, you start really. I mean, Copernicus didn't have much scientific to say, but he sets a path. I'm not saying that I'm the John Baptist of the cell, but I say because I'm gonna finish with my head cut. But I think, I think I'm just trying to call the attention of what that there is a reality, and it's a fascinating reality. I think what I find exciting about science is that things that we cannot explain, things that tease us.
Of course, yeah.
And I think the development of an organism has a lot of fantastic things, as I describe in the book. You can imagine the fantastic view of Spemann and Mangold, where they take a bit of the frog, they put it on the other side, and they get a Siamese twin. I mean, that's magic. I mean, or my favorite is Hans Risch. I mean, the great experimental embryologist of the late 19th century, where he separates the two cells of a two-cell sea urchin embryo and gets two smaller embryos. I mean, he goes berserk. In fact, Risch is a fantastic character because he tries to reason what kind of a machine can do that. And in fact, when you read his descriptions, you see that he's describing a mid-20th century cell. But he doesn't believe that such thing can exist. So he founded the German Parapsychological Society. He became a vitalist. He explained everything because he couldn't believe. And yet there was something there that is fantastic. So this is what I'm trying to say, that highlighting a number of observations that are very difficult to understand simply with genes. There is another reality, and I find it exciting. Together, I mean, we've been talking at a very different level. People don't want to be bored with the details. As I was just saying, I was contradicting myself, but there are details coming, and it's very, very exciting.
Yeah, I totally agree. I absolutely agree. I have two final questions. One's a kind of big one, but you can take it any way you want. The first question is, normal cells, as you mentioned, don't live forever, but some cancerous cells seem to have a longer life. And obviously, cancer in oncology is a huge topic and all that stuff. But from your vantage point, from cells, and I talked to Siddhartha as well when he had the Song of the Cell come out. It was a great conversation, and obviously he's written about cancer. But from your perspective, why? I mean, I know so many people, especially recently, that have been diagnosed with cancer, and it's still a mystery to us. We're still trying to figure out what is it about cancerous cells that are so frustratingly difficult to find, how they're completely working, and then how to make them not kill us. What is it from your vantage point? How do you see this?
Well, I'm not a cancer biologist. I think you mentioned Siddhartha. His book, The Emperor of All Maladies, is fantastic. Great. I recommend everybody to read that book, even if you are lucky enough not to have had a brush with cancer. I think that will give you an insight into a lot of stuff. So making that preamble that I'm not a cancer biologist, I think that those are cells, as we said at the beginning, that have broken this Faustian pact with the organism. And now they are by themselves looking for ways of projecting themselves into eternity, And in a way, the story of Henrietta Lacks, which you know very, very well, it's a very good example of that, because today, there are hundreds of different cell lines all around the world derived from the original one. None of them is like the original one. And I think this is one of the devices that cancer cells use in their quest to remain in eternity, that they have to change all the time. They have to, and that again goes to the heart of the matter, that we don't understand how cells can do that, even at the level of a cancer cell. I think also there is no, and Siddhartha explains that very well, and you obviously know, there is no one cancer. Right. It's like there is no one type of cold. Right. There is, in a way, is the same thing. And because these are strategies that cells that have gone off in a tangent are trying to launch themselves into the future. So very limited answer to your question, but that's all I can do.
No, that's good. The final question is how does
do I get something at the end if I answer it? Do I get a prize? The way you put it, I'm hoping I get a prize.
Hopefully the prize is everyone goes and buys your book. So that's my hope. Okay. How does a cell's eye view of biology help us to have a more complete and robust picture of biology instead of genes, and maybe connected to that is how do you want people to receive your book, most accurately?
Well, I think if I spark their imagination, if I spark their curiosity, if I make them see things that they haven't thought about themselves and the world around them, that will be quite a good thing for the young people. Like many of us, it was a book that we read when we were young that led us to the path that led us to today. Yeah. So I hope that I raise questions that actually stimulate their minds and make them go out and see that. How does it help us see ourselves? Well, there is something that two things that are two simple facts. You and I were never a gene, but you and I were once one cell in the womb of your mother and my mother. So that already should make you think that maybe there is a lot more to life than genes. The other thing is that I'm looking at you and I'm not looking at your genes, I'm looking at your cells. That's what you're looking at me. We are a conglomerate of cells. As I said, the cell, I would say, is the great unknown of the 21st century. And I think this book, I hope it will make people realize that it is worth knowing about these fundamental units that make us be who we are, how we think. If you think about it, the reason why we think is not because genes make us think, it's because there is this network of neurons which are actually firing and interacting. There is more to the activity of cells than the brain, and this is what I try to put in the book.
Yeah, that's wonderful. Well, the book is called The Master Builder, How the New Science of the Cell is Rewriting the Story of Life. It's through BASIC, and it's absolutely fantastic, and everyone should go get it. Alfonso, thank you so much. This was a fabulous conversation, and I really, really enjoyed it, and I can't say enough thanks for you.
No, thank you.